So as I said, I'll start sharing my screen while we wait for other important attendees to join in. And so in general, I thought that I would like to very briefly and in an extremely incomplete manner touch upon some of the underlying physics that governs our study of cardiovascular pathophysiology, how we describe the biology. Now, everyone might appreciate that a large part of the audience are undergraduate and postgraduates. And so what I'm about to present, either starting now or at 4 o'clock, is targeted towards this audience. So Prof. Christopher Mcintyre, who is the leading nephrologist in this town, has just joined us. Hello, Professor Mcintyre.
Hi, guys.
Prof, would you care to say a few words to our students here, please?
The gathered masses. Oh, you did. I didn't know you needed me to say anything right now. Welcome, everybody. This is Sanjay show very much. I think it's the first chance to pull together a whole load of outputs from this multi-modal, multi-organ business that we're all engaged in. And I think Sanjay's the guy driving it, so I'm going to leave it to him.
Thank you, Prof. So in the interests of time, people, would Jay have any objection to us proceeding to my talk, which was scheduled to start at 4 o'clock? How about we start now?
That sounds good, sir.
And therefore,
I need permission to record first, though.
Sure. You have permission to record now?
Okay, I'll start my recording.
Thank you, Jay. So here's me starting the presentation, which was scheduled for 4 o'clock. So Keith, Dr. Goldman, Prof. Frisbee, and Prof. Mcintyre, as I was saying, this description of underlying physics of the biology that we are interested in is very much targeted at the collection of people that we have here. And I present my apologies in advance in case this is ever so marginally boring to anyone here. And so I will present twice. The first presentation is now, while the second presentation is on Sunday afternoon, just before Professor Goldman's presentation about oxygen and such like. And so today, I thought I would like to gloss over a few equations that we use to describe the physics of biology, of the biology that we're interested in. As I said, and I will repeat, this is in no way complete. Neither is it comprehensive. However, I hope that what I show you today induces a little bit of an adventure spirit in you. It induces a little bit of a discovery for you. And those are the intentions of these few slides over here. And so the overview is that, again, what is the physics that we use and that we need? As you can see, as you may see, my take on the definition of physics may be marginally different from everyone else's definition of physics. And on Sunday, we might get to know a couple of formulas, especially for numerical methods of the physics that we will discuss today. And so that's the overview of both my presentations. So today it is physics, and on Sunday, a few numerical methods, rather mention of a few numerical methods. We will not work through the formulas. But fine. And so to repeat the purpose, I hope that today sort of motivates everyone to look for the equations, the formulas, the mathematical descriptions of what is relevant to your projects, to your theses, to your papers, to your project reports, to your technical reports, to the commentary that you write in your advanced codes. I hope that you are provided with some ignition and some background on how the underlying solvers, on how the underlying equations work, what might be underneath the hood. Again, this is by no means a complete description. You should be able to place the solvers that you use. Lawrence uses 1D solvers, Xinyi Wang uses 3D solvers, Tim uses all kinds of solvers, and Jeremiah uses solvers as well as Paraview, VTK. And so we should be able to put our understanding of how these solvers work as advanced informed users and put all of that into a wider context of the machinery that is underneath the tools of the trade provided. And the most important sort of hope is that our discussion today, and it has to be a discussion, feel free to ask questions, is to induce inquiry as well as exploration. And I sincerely believe that that is the best way to learn. To suffer and to do it firsthand is the best way to learn. Okay? And so that is the purpose of today's talks. And so without further ado, I will get started with the equations and formulas. And I hope everyone bears with me because I have been afforded an extra half an hour. So I might take it a little bit slower unless I decide to completely forget the content. And so the first thing I thought I would like to mention here is something that we've done quite a lot over the last couple of years. We've had recordings from smooth muscle cells. We've had the lengths and morphometry of large blood vessels and small blood vessels as they go from the aorta to the brain, back and forth to the kidneys and whatnots. Some of us, I think Riva Patel, have also started working with epidemiological equations. In terms of what Jeremiah is busy with these days, as shown in one of the gray boxes on the left-hand side, he is right now concerned with appropriately placing his cardiac function in terms of forcing terms, elastances. And quite importantly, as we make these biophysically detailed models, every piece of work that we do will have sensitivity analysis, will have uncertainty analysis, and we will have to justify this against all the wet lab work that I so hope that all of us get involved in. And to do all of these things, firstly, we have to know the formula represented by this F with an arrow on its head. The arrow represents something that means F is a vector function. It has several components. It may not be just one formula. We have to be double guessing, triple guessing many of these formulas. Assuming that we do come to a consensus on what the formulas could be, and our first and second years did this with Professor Jeff Wild a few months ago. We are then faced with the problem of working out what the parameters could be. parameters are represented by P over here with an arrow meaning that P has multiple components, P1, P2, P3, every single one of which needs to be estimated, either driven by data or by intelligent guesstimation. And so data fitting to known formulas, and we're still working out what formula to fit your data to, is a significant concern of ours. And the functions that, the Fs that we work with these days is something called a sigmoidal that J is concerned with, fitting of the L-type calcium channel to experimental data to sigmoidals. They fit steady state curves to sigmoidals. I believe Randa is also busy with similar fitting for her PTSD, Timothy syndrome problem. Lawrence has fitted data using B-splines, Bezier curves. Why just Bezier curves? Even Bezier volumes to model anatomies, to generate aorta brain complexes, which I hope he will discuss tomorrow or day after. And Riva has explored epidemiological equations, which are essentially taking functions that we think fit the data and then representing clinical epidemiology by setting parameters there. So, we will come across the use of data fitting in many different ongoing projects. Now, the algorithms that we have tools to work with. My favorite is called Levenberg-Marquardt. Another favorite of mine is also called Neldamead, something that I used with the bioengineers in the University of Bologna in Italy. I worked with the Manchester Institute of Biosciences to implement something called a weighted steepest gradient method. But in recent times, it is becoming more and more obvious that all of these are compute-intensive algorithms, and we should move on to more clever ways of using our time as well as the supercomputer's time. And so simulated annealing is on the cards. Monte Carlo methods are on the cards. And I'm happy to announce that we do not have to start from scratch for most of these algorithms. Some of them have been implemented by yours truly, but quite a lot are already available in very straightforward codes, such as GNU-plot. MATLAB has extensive resources for all of these methods and then some. The same is reproduced in Python libraries. And all we have to do is train ourselves on the prudent use of these tools that are already available. And so data fitting is a very sort of early step in the modeling process, at least in the current projects. Data fitting everywhere. Now fine, once we have fitted the data, I thought I would like to take a digression over here. And the digression is to call especially people who are working with this large number of files. They might be running very many simulations at the same time on different models at the same time. As we have seen recently, we generated a new description of the human circulation, January gone, which is now under revision. Right now, the revision goes on. And it hit me that we should make some effort to avoid technical debt and maintain electronic hygiene. So I would really, really, really appreciate it if every single one of us, clinician or not, bioengineer or not, mathematician or not, adopts a technical debt-free approach and a hygienic approach. And by technical debt, what I mean is that we have to diligently spend enough time on your platform to phase in all sorts of strengths. Because if not, we are then digging ourselves into a hole as shown on the right-hand side, where literally rubbish accumulates behind us and there's nowhere to go home at the end of the day. And so if we invest ourselves prudently. This does not mean working 24/7. This means working effectively. And so by adopting principles of appropriate working, we should avoid technical debt. Okay, do not adopt methods that involve convenience. Do not just do it so that you can move on. Please do invest yourselves to the point where you have to have anticipated upcoming problems, upcoming comments by other experts who have not worked closely with you or me or with all of us. And the products I allude to as far as technical debt are concerned, firstly, are the platforms, because these are the things that are going to live for several years, maybe even a couple of decades with us, okay, 2 or 3 decades. We have to implement technical debt in our manuscripts, in the expectation of what comments to expect from both internal as well as external review. We have to reduce or minimize our technical debt in our research results, in our essays, and in our data analysis. Now, in case anyone is interested, there is a very elegant group run by Philippe Cruchton in UBC, and they are right now starting a brand new effort towards sort of laying down the principles of how to minimize you cannot eliminate technical debt, but you can try to minimize your technical debt by appropriate pre-planning and implementing in a robust manner, excuse me, sorry. Following that, a close cousin of technical debt is documentary hygiene. I very much, we have had conversations one-to-one, as well as across sort of in communal meetings, where I hope everyone organizes your electronic materials, your documents, your files, your outputs, your inputs, your outputs, what goes where, especially when life gets really busy and you're running about half a dozen, even 2 models. If you're running 2 models at the same time, they produce terabytes of data. So you have to be, I hope that everyone adopts principles of documentary hygiene, okay? And documentary hygiene then applies to writing documents, to writing manuscripts. If you write a manuscript, say, for example, a good principle is to develop your manuscript such that it then cascades into thesis content and such like, okay? If you've made figures and ran simulations for this workshop, you may also sort of do a little bit more work so that the same material is useful to you in the future. And finally, if we bear some conscience, we might also have social hygiene. So this was a digression about technical debt that I thought might be helpful for how we progress our respective projects. Coming back to track, we've discussed the use of bog standard functions that are fitted to data. Often experimental observations, say for example flows or strain rates, are essentially derivatives of underlying state variables. And the description to start on this slide would be something that is called an ordinary differential equation. A exceedingly elegant book to take ordinary differential equations lessons is a book by Berkoff and Carlo Rota. Exceedingly elegant. That's what I used many summers ago, and winters ago. And ordinary differential equations these days are used by us for our reduced order hemodynamic modeling, something that maybe Tim might touch upon, if not. Cardiomyocytes, we describe cardiomyocytes using ordinary differential equations. That is a concern of Randa. About intracellular calcium processes. Calcium sticks to everything. Calcium is a very important second messenger. And so we describe calcium-induced calcium release inside of smooth muscle cells, cardiomyocytes, using ordinary differential equations. Jeremiah and Tim have already finished describing baroreflex phenomena or some sort of feedback mechanisms in reduced order whole body vasculature. The autoregulation is described as a first step using ordinary differential equations. Our current descriptions of smooth muscle cells also, as well as proteins that J is concerned, are described by ordinary differential equations. And the work over here is again the same thing, in that work out the appropriate function. That in some sense is a deeper problem, but there may be some legacy. Say, for example, Hodgkin and Huxley, or maybe Timothy Seacombe or Dr. Goldman have written some formulas that we can take as sort of established work. And in that paradigm, we fit the functions to your data by estimating the parameters, followed by solving your ordinary differential equations with any which given initial conditions. So your pathophysiologically relevant initial conditions will dictate the solution. We have some very nice ways of solving these ordinary differential equations, something that we will discuss on Sunday afternoon. And furthermore, because ordinary differential equations are that much easier to handle than something called partial differential equations. See, I've written D over here. In a minute, it might become a curly D. And so if our descriptions to start with are reduced order, these models are quite amenable to uncertainty quantification, to sensitivity analysis, and everything in between. And for those purposes, we have tools to implement very good random number generators using Medicine Twister. We can sample from the space, from the hyperspace of our parameters using latent hypercube sampling, reduces compute requirements considerably. In recent times, methods developed by Richard Clayton in Sheffield and by someone else in Exeter called Gaussian processes can also let us generate populations of virtual patients, reduced order virtual patients, using which we can perhaps, in the very least, generate an expectation of what might happen when you apply a treatment. And importantly, give us a sense of what to expect, say, for example, in spatially extended systems. And so unless something drastic happens, a key feature of our progress will be to model pathophysiological systems as reduced order systems before we go to spatially extended systems. We have now started going to spatially extended systems. But ODs form a robust part of the workflow over here. So that was ordinary differential equations. However, biology is not also simple. And oftentimes, biology has memory. So there is a delay in response of, say, a blood vessel to a stimulus. And that could be described in many, many different ways. But one way would be a delay differential equation, which has a delay represented by this time tau. And so in that case, rather than having just one initial condition, we have an initial condition till up to tau time units in the history. And this is useful both in image processing in Dr. Tingley's lab in robots. It's also useful in our descriptions of hemodynamics, cardiomyocytes, tissues, and such like. The algorithms, I believe, were discussed a while ago. And if not, we will touch upon this algorithm again on Sunday, unless we were. So I have forgotten if the algorithm is there or not on Sunday. Now, fine. And so moving on to slightly more interesting ways of describing physics. And this would be the simplest way of describing physics, describing some biology in physics, some biology using physics, would be the diffusion equation as shown at the top, in the top row over here. Written very simplistically, you could have a variable, and its diffusion then is described by either being equal to nothing or any formula that you want. Okay. So for simplicity, I've written 0 over there, but you could have any other constant right-hand side. Right-hand side is anything after the equal to sign. Slightly more interestingly, something that Lawrence is interested in, something that Freewa is interested in, Tim is interested in this, as well as Randa, in that cardiac fibers, cerebral fibers, are often described using fiber directions, fiber orientation. And that is imaging driven, summarized by this tensor, a n by n matrix represented by this D with a two-sided arrow on it. I could not find any other way to represent a tensor there, but fine, there you have it. So the D then represents your summary of fiber directions, either in the heart or in the brain or elsewhere, even in striated muscle perhaps. And roughly about those are the directions that blood vessels are also oriented. And so just as a part of explanation written in red is the generalization of the spatial derivative, pronounced either as nabla or as gradient or as del. You can call it whatever you want. But it takes the gradient in a given direction or in several given directions, and therefore is a vector operation. But fine. So diffusion is used by many groups in Western as well as elsewhere for image segmentation. So the Khan lab uses diffusion quite a lot. The other groups, or also the same group, we use diffusion for edge detection and image segmentation, as well as to describe image texture. So there is a measure called Fractal Dimension that we found could be applied to clinically obtained CT data. So we could apply, and that involves this operator. And more biophysically interesting is that imaging often gives us some information about how cerebral neurons are oriented, about how cardiomyocytes are oriented in only a few different locations. Typically, that would be some form of diffusion-weighted imaging, MRI imaging. And using that, I'm happy to announce that we have the anisotropic diffusion operator that can then complete the description of fibers in hearts and brains, so that we can then explore the biophysics in those organs and other organs furthermore. And so the algorithms that we will discuss this a bit more come Sunday, involve pseudo-time stepping till convergence is reached. We use finite differences, higher order finite differences, at least inside the domain, inside the brain. I don't know about when the brain comes in contact with, say, the cerebrospinal fluid, but definitely inside the brain, we are higher order finite differences. However, I would like for everyone to explore other methods such as spectral methods, such as finite elements. And there's a long list of other methods that I hope you come across in your self-discovery. Now, to start understanding how to describe physics, how to use maths to describe physics of biology, I highly, thoroughly recommend a very classical book by Lev Davidovich Landau and Lifshitz. Summarily, nominally, it's called Mechanics, but the main training that books like this provide is the use of the very sort of clever use, the intelligent use of equations, the simplest of which is written in front of us at the top here. And so the diffusion equation, in summary, the diffusion equation is extensively used by several of our platforms, as well as the dynamics described in quite a few of our platforms. Diffusion equation, everyone. But fine, so that was the diffusion equation. Generalizing the diffusion equation, we can add a term. So written in purple here is what we discussed on the previous slide, the diffusion of some quantity C in space. This is something that we did discuss on the previous slide. However, we can generalize this somewhat and add a reaction term, which means that if you're sat at some place in the brain, say inside an arteriole, or if you're sat at some place inside the heart, say at a cardiomyocyte location, both of these things have ionic and protein currents, transports, going on back and forth through the bilipid membrane. And those processes are summarized by this term I that I choose to call current. It does not have to be current. It needs to be just some reaction. Now, written as I've written at the top over here, the description of electrical waves, of calcium waves, of metabolic waves in our organs could be described by reaction, what is written in red, and diffusion, and the sum of that for several of these biological phenomena that we're interested in. Of course, this takes initial conditions. Now, written in this way, I would classify this equation as something that I would call a homogenized description. And by that, what I mean is that the purple term will cede to your efforts as far away from the X where you're sat at. So a cardiomyocyte sat at infinitely long, far away distance from a brain neuron does get affected by each other. They do get affected by each other, given our assumption of homogenization here. But such a reaction diffusion description still holds, will be used for the near future to describe cardiac waves, to describe signal propagation in arterioles, to describe calcium, intracellular calcium communication, to describe experimental measurements that I hope many of us do, to model microvasculature, say, for example, under ischemic stroke. Yeah. So reaction diffusion is a very, very useful concept. But if you are feeling adventurous, and if you strongly believe that cardiomyocytes are only connected to their nearest neighbors, say, for example, by gap junctions, by some other mechanical proteins, then you might modify your current reaction diffusion description and write the second row of equation here, which then becomes a very large set of ordinary differential equations, where the J are all the cells that are only in your surrounding, either in 2D or 3D or even in 1D. And so written at the top is a globally coupled system. It's a homogenization approximation. I will use it for forever. But if you fancy making your system locally coupled, feel free to have your gap junctional and IP3 coupling for nearest neighbors. The oscillators can be coupled to nearest neighbors. And the algorithms that we use to solve, especially this reaction diffusion system, is something that we will discuss to some depth on Sunday. And this follows a sequence of operator splitting. You split the purple part and the red part. You use your very efficient fourth order implicit finite difference. And if you're feeling adventurous, you can then do this as a multi-step process. Lots of libraries such as Trilineos, Phoenix, Petsky. Once you dig into these libraries, they will very beautifully tell you exactly how to do all these efficient calculations. Going back to reaction and diffusion also describes how Alfie's spots appeared. J.D. Murray, who I believe was either in Warwick or elsewhere, has provided extensive description on how reaction diffusion can be used to describe lots of ecology, lots of evolution, in addition to movement of electrical waves from one place to the other. J.D. Murray's mathematical biology is thoroughly recommended for a guttural understanding of all things reaction diffusion. This is not the only book, but this is one of the books that I do quite like. But fine, coming close to what Mia Wang, Lawrence, Tim, Jeremiah, and everyone in between are working with is the flow of blood through large blood vessels. And the solver that is now provided to all PM3 users and whoever wants it. So whoever wants it has access to this solver. It's literally spelled as SV capital S solver. It's literally spelled as that. And this thing provides numerical solutions to the biophysical description given by Navier-Stokes and its cousins. Okay, and so the box standard fluid flow, where U is the speed or the velocity of the fluid, and P is the pressure. P is always a scalar. It may be a scalar field, but it's always a scalar. You can take its spatial derivative and make it a vector, but on its own, it's a scalar. It's a scalar field. On the other hand, stress is something, a mathematical entity called a tensor. What we learned several months ago, people, is that tau is a matrix with many columns and many rows. And that is how both stresses and strains are described as tensors. And so the description of how fluid flows is given to you by Navier-Stokes. For completeness, there has to be continuity. We will come back to this eventually, maybe today, maybe on Sunday. And all the things that SV solver output gives you, typically as a large number of text files or as summary VTK outputs, we obtain, say, for example, the wall shear stress. We obtain the Reynolds number and several other quantities. And all of this is obtained using additional equations and shown over here with tau, the tensor, obtained using I forgot now, but I think mu is the viscosity, and obtained using a suitable sum of the derivatives of the velocity profiles. So all of this then is provided to us by SV solver. So these are the equations that underlie the numerical solution that we have here. I may have forgotten to write a couple of terms in any one or all of these three equations. For that, I apologize. Making combinations of all these U's, P's, taus, rhos, and mu's is what gives us the clinically measurable quantities. So we have to prudently combine all of this and come up with a number that can also be measured in the clinic. And that is the expertise that I believe several of us are generating right now. Okay, fine. So the platform available on our local and national supercomputers is your 1D solver, is your 3D solver, is your 3D solver with fluid structure interaction. Yeah, fine, that's okay. But to gutturally engage, I hope that I sincerely ask for those who are willing to themselves implement the 1D, 2D, and 3D equations of fluid flow and all related equations. You can do that in MATLAB, you can do that in Python, and I have no problem everyone using advanced libraries for these implementations. It's a very enjoyable learning curve, I'll remind you. Now, to get used to this symbolic notation, these massive dots and these upside down triangles and these arrows and whatnots, there's an exceedingly elegant book about electrodynamics, classical electrodynamics, by this guy, J.D. Jackson. This is a classical textbook, and on the inside cover of this textbook are written all these equations, and that might then get us started in our journey of exploration. So Navier-Stokes equations, everyone, in 3D. We have the solutions in working form on all our computers, but these may be some of the equations that govern the solutions we obtain. So we have to be very careful about how we use these solvers if we want these equations to do what you want them to do. Navier-Stokes. Now, moving on to something slightly different from fluid flow. This is something that has significant IP value, significant knowledge value, is the calculation of a stress followed by strains. So on the left-hand side, I've shown the partial differential equation that you can use for calculating strains. If you know the displacements, you are the displacements, the deformations. And if you make a suitable combination of the derivatives there, you might end up with a whole bunch of strains. If, say, AI, a subscript, is repeated, in the same formula, a summation is implied. So this term here stands on its own, okay? But a component of your strain matrix could be worked out, is worked out in this way. Written as I have written on the left-hand side equation, something that I'm pointing to with my arrow. This would be called the engineering strain. Because engineers are such clever people, they define strains in many, many different ways. There's a definition of strain using called Kharche strain, and so on and so forth. Engineering strain. But fine, the relevant part of, not the relevant part, but the clinically measurable way in which we deduce strain is what is written on the right-hand side. So on the left-hand side of the equal to sign is something called the strain rate. And so this can be directly measured either from dense cine MRI imaging, or from echo imaging, or from some other kind of imaging. And on the right-hand side is its relationship to the strains that the respective tissue experiences. And so this together might be a description of how we study strain, of how we quantify strain, and these are the governing principles there. equations for strain that may or may not be implemented in dense analysis that Yusuf and Daniel will speak about. So here's two photographs I found. One is on the right-hand side, so of the cerebral vasculature. There are pile regions apparently. So there are these large arteries that run along the grooves of white and gray matter. And every so often, they put down roots into the white and gray matter. That gives rise to vasculature beds, as shown towards the bottom of the left-hand side figure. And so I wonder how you might use all the equations that we've spoken about so far, fitting of data, of Bezier curves, of partial differential equations, of ordinary differential equations, of autoregulation, and how you would apply it to here. How would you describe the delivery of oxygen by this vasculature to the white and gray matter, which is not shown in this diagram? In contrast to cerebral vasculature, which may or may not look like this, lots of tubes with smaller tubes falling into the tissue. Yeah. On the right-hand side is shown a caricature of uterine arteries just before parturition, yeah, just before childbirth, in that the main arteries here, something called arcuate arteries here, give rise to smaller feeding arteries, and to maximize that trajectory in the endometrium, they become spirals. This is just before parturition. This has been studied by Dr. Meyer Zamir in Western University Maths. Yeah. And we should find sort of the equations, the descriptions of blood flow, of oxygen delivery, of metabolites delivery under all these different kinds of anatomical conditions. There's a whole bunch of biophysics and physiology that has not been shown in these two diagrams. But I thought these are interesting diagrams. But fine. The next thing that I'm really concerned with these days is the working of the principles of liquid flow through porous material. Now, Darcy's law has been used to describe groundwater, of how water seeps underneath Fanshawe Dam or through Fanshawe Dam walls and gets to the other side. And this set of partial differential equations describe pressure in your tissue or in Fanshawe Dam's bed. Okay, it describes pressure. It describes the dynamic flow rate. Now, this is not the flow rate, but rather it is a dynamic flow rate. We have to do something to this vector v before we can get the actual flow rate. Okay, and using this gradient equation, as well as a couple of its sister concerns, we can then describe the movement of drugs inside of kidneys and livers. Yeah, we can also try to study ischemic stroke microvasculature and how it may get perfused. Say, for example, after the clinician pulls the clot out of the blocking vessel. Does the microvasculature even get enough blood after treatment? And does it get blood soon enough to regain healthy status? It can also describe the movement of groundwater. And now, although yours truly is busy implementing this in one of our platforms, I would be thankful if someone takes it upon themselves to implement this either in Python or MATLAB and make meaningful simulations. This becomes an alternative to a slightly opaque library-based C implementation. in MATLAB and Python, it may not appear to be so daunting, and it may be easily uptaken by other users who may not be interested in the workings of any software. So that is one of the challenges that I can see that we should address as a mini project, as a side project, and that will be exceedingly useful for several months, years to come. And so this is an equation that we will use in the near future. But fine, an important equation is given microvasculature, given the brain and it has microvasculature, given the heart and it has microvasculature, given muscle, striated muscle. A relevant question is, how does stuff go from the microvasculature into the tissue that uses that stuff? And after stuff is used up, how does the rejected stuff come back into the microvasculature so that it can be ejected from the body? This is elegantly described by two people. One is Dr. Alexander Popper, and the other one is our Dr. Daniel Goldman. Summarily, this equation is called the Popper-Goldman partial differential equation. The reference is given at the bottom of the slide in case anyone is interested to find equations 15 and 16. But even if you don't find that reference, I hope that Dr. Goldman explains this and then some to us on Sunday afternoon. So please do attend the session on Sunday afternoon. A thing that I would like to draw everyone's attention to, especially those who are really, really busy implementing simvascular scripts to simulate 3D fluid flow, 1D fluid flow, all kinds of fluid flow, is the requirement for boundary conditions, appropriate boundary conditions. Now shown at the top left-hand corner is a extremely sort of horribly written summary of what a partial differential equation could look like, written with an upside down capital V called lambda, and it operates on several variables summarized by U. And if you write it nicely enough, it describes the physics inside the region omega. Now the region omega is shown on the top right-hand side on a structured grid. So I have a structured grid. You can see the boxes there, structured grid. Solutions are obtained at the place where the vertical and horizontal lines intersect if you program this as finite differences. Yeah? But whatever is inside over here of this uneven shape is my region, that's my internal region. Every region has a boundary, and it is represented by a D before the omega. So this symbol here is pronounced as omega. And if you write a D before that, what you mean is that consider the boundary over here. Now, quite often you are also interested in the normal to the boundary at every location. I've shown the normal to the boundary at one location, yeah? But it is exceedingly useful in many different ways to know the normal to the boundary at all locations, especially where your solution must exist. Yeah. And based on the boundary conditions that we program into our simulation scripts, the underlying ethos is summarized in the bottom row over here. So one family of boundary conditions are called Dirichlet boundary conditions. So this is Dirichlet. You know, he's looking elsewhere. So they're making a picture of him. He's looking elsewhere. Dirichlet. What Dirichlet thought was that if you take the variable on its own, in our case, say for example pressure or the fluid flow, and he tells us that if you assign a value for your variable of concern at the boundary, couple that with the description of your physics, the ordinary or differential equations that you're using, you will have a sensible simulation. You will have a sensible solution. Okay, so those are your Dirichlet conditions. So Dirichlet conditions say that the value of your state variable is given at the boundary. Yeah. But fine. So Neumann, this is Neumann. He had sort of Einstein-y looking hair. So that's Neumann there. So what Neumann goes is he goes take the normal and take the gradient of your solution. Yeah. And you are ever so slightly inside your boundary in the domain to be able to do this. You're ever so slightly inside. If your life is a total misery, you might also wish to consider the fiber directions in the heart and the brain, and combine all of this as this dot product on the left-hand side of the equal to sign. And if that value is given at every location of this boundary, then the solution of your partial differential equation denoted by this lambda here will be sensible, may be sensible. Okay. The third family of boundary conditions that you're interested in is given by Robin. This, I think, they tell me this was Robin. So he's holding something there. He's showing me something. He also had some elegant waistcoats. And what Robin tells us is that you combine your Dirichlet boundary conditions and your Neumann boundary conditions into the same equation. These are not two separate equations. You take so a little piece of your boundary condition is Dirichlet, a little piece of your boundary condition is Neumann, and together that one equation quite often ends up as an implicit equation, is your Robin boundary condition. And all of these occur in the simvascular scripts that you are right now making to do all your aorta and your coronary and your brain simulations. So they come up. But fine, Mr. Kharche, so this is Mr. Kharche, and he is much more cleverer. And he figured that we don't need to describe things in this manner. As long as we have one equation for Dirichlet and one for Neumann, that will also be sufficient description of boundary conditions to give us a sensible solution to this partial differential equation, to this sum equation, to this equation. So these are the boundary conditions that come up in the scripts that everyone is either generating using other scripts or are writing painstakingly using text editors. These are some of the boundary conditions that we have to be concerned with. The underlying sort of process. Always pay attention to a lot of the boundary conditions. Boundary conditions in our world may often be called a microvascular bed attached to the end of a terminal of a vessel, and that sort of thing. But fine. So do pay attention to boundary conditions. Otherwise, our solutions are nonsensical. So here comes sort of on the image I came across that shows the exceedingly different architecture in the liver, in the retina, in the brain, and in the lungs. And Gabrielle is going to teach us how to do architectomics with a certain kind of vasculature. A description, a stained image of several vasculatures people. And so coming back to boundary conditions, this is a talk I heard recently by someone called Samir Parekh from Houston Heart Center, who is a nephrologist. And his focus, or rather what he's saying, is that acute kidney injury is when there is too much fat surrounding the cells or the structures, the glomeruli in your kidneys, in the patient's kidneys. Shown on the left-hand side is a healthy kidney stained with something. And on the right-hand side is an unhealthy kidney stained with, I believe, Masson's trichrome. And the Masson's trichrome goes and stains the fat as red. And so this is a kind of intricate geometry, omega, that you have where you have to go and apply your boundary conditions also prudently before you can let loose your dynamic simulations, which then might produce sensible results, sensible indications of how the system progresses. So I thought this might be a sort of interest inducing image. And so the next thing on my mind is sensitivity analysis, is uncertainty quantification. And this is in some ways going to be described by Jay eventually. And the methods that we have right now in the bag are called partial rank correlation coefficients. So these are essentially linearized regression fits to any data that you throw at us. And the code or the platform can then give you this PRCC, partial rank correlation coefficient, which is essentially a ratio of the covariance of certain residuals and the variances of those said residuals. But without going into too much detail over here, working out of sensitivity, of sensibility of models, and of uncertainty is crucial to our work, because our biophysically motivated models will never be completely identified using experimental or clinical data. There will always be free parameters, which then we have to justify. We have to robustly assess what values we should assign them and how uncertain we can be in the values that we assign to them and still get reasonable outcomes from our computer modeling. So this is a bit of tool that has developed over the years, and using partial rank correlation coefficient or its very close cousin called mutual information estimator, we can then develop predictors as shown on the left-hand side over here, wherein if you give a whole bunch of observables, say for example, heart rate, blood pressure, blood works, yeah, and if then you have trained your model sufficiently, and training essentially means working out the values of these AIJs here, of these regression coefficients. And if you've done that, you then quickly go and do this matrix multiplication to this vector, and you can generate predictions. And Riva is right now concerned about predictions in acute kidney injury as well as ischemic stroke. But shown on the right-hand side here is an even more extensive description of how large data can be handled, how your learning programs should be used. Yeah, but be careful. Not everything is useful for everyone's purposes. So be very careful of picking ready-to-use tools. It is better for us to develop our own tools to a certain extent, so that we know what goes into that. A couple of figures I saw at the National Art Gallery in Trafalgar Square a couple of years ago. And the way I choose to read these beautiful, beautiful murals, paintings on the floor, is to have a sense of humor and to think disruptively. Rather beautiful things. They are right on the floor. So a comment on large data and related. I'm coming to the end of this part of my presentation in a minute. But before I finish in a few slides, I would like to thoroughly sort of remind everyone that not everything artificial intelligence is either artificial nor intelligent. the person getting paid for running the artificial intelligence section of Microsoft, called Kate Crawford, goes, artificial intelligence is neither artificial nor intelligent, as written at the bottom. Someone on social media summarized sort of the warning that we should sort of bear in mind, is that even before the powers of artificial intelligence, of exceedingly high microscopy, resolution microscopy, of omics, of engineering, the most valuable resource that we have is the researcher's skill, and their skill to carefully design and execute experiments, to know how to deploy a proper workflow. And people make no mistake, this very much applies to your computing lark. So this is not just something that the clinical experimentalists bear in mind. There's not just the experimentalist who bears this in mind. We also have to remember all of this. So brief comment. And so in summary, I would like to end by saying that, by admitting that cardiovascular pathophysiology is exceedingly rich. It's exceedingly stimulating. And we have many, many, many things to study here, both for the basic scientist, for the clinical scientist, as well as for the translational scientists. And so it is rich in phenomena. It's exceedingly stimulating. By definition, cardiovascular physiology is something that is multi-physics. So you have electricity and blood flow. You have blood flow and metabolism going on. You have blood flow as well as elasticity and biomechanics going on. And thankfully, we have a little bit of maths, but you are not just looking to sort of do a little bit of multi-physics, structure, fluid interaction, and so on. That, I hope, is not our life goal here. Our efforts must stick, as other leaders show, to improving society. And in our case, a useful aim to bear in mind would be that within our lifetimes, you want to improve the quality of life of critically ill patients, improve patient outcomes, not as a legacy that you leave for the next person, but within your lifetime. Okay. And so as far as the algorithms for this summary are concerned, please do talk to many, 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 many people. Keep your eyes and ears open. Tread with care, but be bold, okay? You have to process all the information that is coming your way. I know this is uncalled for advice, but fine. For what it's worth, I'm dishing it out, okay? And so with that summary, I have a question for you now. Why do you want to become, most of today's attendees, I believe, want to go and become doctors?